Assalamu alaikum viewers, welcome to my channel Genome Wide Study. If you are new on my channel, please subscribe and press the bell icon so you will get the notification of all the new videos. In this video, we will talk about homology modeling. But first we know that what is homology modeling. Homology modeling is the construction of an atomic resolution model of a target protein, the target protein on which we are working, from its amino acid sequence and an experimental three-dimensional structure of a related homologous protein template. It means that we will predict the three-dimensional structure of our protein on the basis of already experimental proved structure of a protein, like it is used to determine the structure of a target protein on which we are working based on the sequence similarity of template protein whose 3D structure was experimentally proved, and its data is available in a protein database bank. But there are some prediction of the structure, that how to predict the structure is accurate. The protein structure prediction on sequence homology base means the sequence identity or similarity of these both proteins is higher than the structure accuracy is also higher. When two proteins have similar sequence, then their structure will also be similar. And third one is when these two proteins, that the protein of the template and the target protein must have greater than 50% similarity in their sequence for structure accuracy, means the cover region. When target template sequence identity targets our protein, while the template protein that whose structure is already proved by experimental, their sequence identity is less than 30%, then the model accuracy is decreases, and then we will didn't take that model. Then how we will do this? So we will do this from our protein amino acid sequence. If we have the protein amino acid sequence, so we can predict or do its three-dimensional structure in two ways. One is the experimental way and other is the theoretical way. Through experimental we do different techniques like X-ray, NMR, mass spectrophotometer, while through theoretical we use the homology modeling, and then we will predict the 3D structure of our protein. And how to do this? We use the Swiss modeling for this homology modeling. How to use the Swiss model? Let's start. Go to Google and write Swiss model and search. You will find the Swiss model.xpc.org link. Open it, and here it gives this window, and you can click on start modeling. So you can enter your protein sequence. So open the file in which you have the amino acid sequence of your protein. Copy the sequence and paste here. When you paste your sequence, it gives different color. It means the this program has accepted your sequence and your sequence is accurate. If there is some error in your sequence, then it gives the white color and its color didn't change. And here you give the program name, project name, like I give the gene 1, and here you can write your email address. If you write the email address, then when it finish the work or modeling, then send the results to you on your email. And then here is a two option: search for template and other is a build model. So first we search for template that our sequence is similar to which model which is already experimentally proved and its data is available in the protein data bank. To search for template, here it is working. It works on the basis of BLAST and alignment, means the software BLAST this sequence in the protein data bank where the experimentally proved sequences are available, and then align the best model and then give us the different template results. And here is the work that it is running is here is a running HHB lights against HTML and like cache query alignment and like this. All this the software is doing the working, so it is mentioned here. So it will take a little bit time. So it gives us about 50 templates, means it gives the 50 models from protein data bank or 50 models whose structure is experimentally proved on the basis of sequence similarity. And here are the some information about these template or models, like here is the name of this gene, that here is a PDB ID, and here is the name or function of this gene, and here is a GMQE value and QSQE value identity, and this is method through which this model was proved through experiment, X-ray method or mass spectrophotometry, or like this. And this is its nature, that it is either monomer or oligomer, and here its legend is present or not. And here you when you click on this, so it will show the coverage and that how much amino acids are covered with this model. So after selecting the model, that which one is your required one or the best one, so you will check. One is its coverage, that how much this model is covered with your protein, and as it is maximum, so it is good. Here is a 0.95, it means it's 95% it covers the your query. Other is the GMQE, Global Model Quality Estimation, and its value is 0 to 1, and as it is higher, so it is good. And this is the identity, and how much your sequence is identical to this one predicted one or this template one, and its identity, as we earlier said, that is about 30%. So it is also good. So you can select these models that have fulfill our requirements. One is this one, and other one is also here is 9.92, mean 92% coverage, and it is 94% coverage, and it gives 93% coverage. And here is the GMQE is also good, and here is the identity also reasonable, and here is the identity is less than 30%, so we didn't consider this model. So you can select these 3 to 4 models and then click on build models. So it is running modeling job 1 2 3 4. So here we have selected 4 models and their results are here. We can see that it's the results of model 3, it's the results of model 1, and here is the result of model 2, while the 4 is still running. So we shall wait a little. Now we have got all the 4 models that we have selected, and on the basis of these models it gives our model prediction and gives some properties. Like here it's a model 3, so it is a sequence identity, and it's a template PDB ID, and it's a description. While here you can see it's a GMQE, global model quality estimation, is also good, but here you can see that Q mean is over -4, which is not good, and here is you can see that the thumb is down. It means that this is not a accurate model, or the accuracy of our sequence and the model is made for on the basis of using this template is not accurate, so we will not consider this model. And here the model 4 is also, its value is q mean is over -5, and it is not good. And here model 2 is also, but when you see the model 1, its sequence identity is also greater than 30%, and its G and QE is also better, and its Q mean is less than -4, and it is acceptable, or its accuracy is good, and its thumb is up. It means that we can use this model. Here are some parameters, like here it gives the alignment. This is our sequence, where this is alignment with this query protein, or the protein whose structure is already determined and present in the protein data bank. Here the blue line shows the accuracy in the model, while the red one presents the not accurate, or accuracy is less. When you select these amino acids, so it will also shown here in this model. Other thing is it's that local quality estimation. These are the presence of residues, and it is little bit very fluctuated one. And other is the comparison with non-redundant set of PDB structure, and it's a Z-score. These dots are a little bit lighter showed the value greater than 2, a little bit brighter showed the less than 2, and the darker one showed the less than 1, and this is our model that it lies here. If you want to save these pictures, so here click on save as PNG and use it into your further project. And here it shows the Q mean value lies. Now you can also assess your structure by Ramachandra plot. So here you see it build the Ramachandra plot, and all the residues fall in the selected areas. I mean, it is a good, and I didn't found any amino acid in the non-selected area. So this model is good. So we can download this by clicking here and download its PDB format. This file we can download it in the PDB format, model 1, save it. And we can also save this the PDB file of our this template protein whose structure is available in the PDB database. And when you click on it, it gives information of this gene, like its method through which it was experimentally proved, its LIGO state at a monomer or a heteromer, its legend, and its experiment who have done this, and it's this one is a citation like this. So we can also download its PDB file by clicking on here. So you can check these PDB file into your any software, like I am using Chimera, so you can open it in the Chimera. File. You can also change by ID, like you have that ID of that your protein file, or if you have the PDB file, then directly open it. Here is the model 1. So this is your protein model that we have built, and here you can also give some color, like here is depiction and color secondary structure, and here you can give any color that you want. That helix you want to give them like this, and a strand, whatever. Anyone that you want to give. So and here you click on apply, it will get this color. If you want to change it, okay, or you want to give them rainbow color, apply, and you will get this color. And now you can save it, save image, homology, gene 1. Here you can select the format, either in which TIFF, JPG, or whether in which, and then save. You can also open your file other, that if you want to see the homology or alignment of your structure, both the structure, so you can also open the other file that you have saved as the your template. So open it, and then you can see its alignment, that this one fine one. So you can also change the color of this to make it. Here you can select this, which one you want to change the, and you can give one color, or whether red one, I think is okay. Right. So here the red one is our template where the rainbow color is our query protein or protein on which we are working. So you can also save it and use it into your manuscript or where you want to use it. So this is our video about homology modeling, how to do and what is homology modeling. Hopefully it will help you. If you have any query, please comment, like. Thank you.